Hi, this is Dr. Rosario, and in this lecture we're going to be talking about eukaryotes. So these are the cells that have nuclei. In this lecture, we will discuss the evolutionary relationship between bacteria, archaea, and our eukaryotes. We'll discuss the different organelles that we find in our eukaryotic cells and the functions of each. I will talk about the differences between prokaryotes and eukaryotes. And then finally, we will discuss some of the major groups of eukaryotic microorganisms. So remember, the name eukaryote comes from the Greek that means true nucleus. All right? And there are several different types of eukaryotes. We are eukaryotes, we are animals. Plants are also eukaryotes, fungi, and then our last group is our protozoa. We're going to focus in this lecture on animals, fungi, and protozoa. We're not going to talk much about plants since they don't play much of a role in human disease. So there are lots of different types of eukaryotes and eukaryotic cells. You can see in this figure, eukaryotic cells come in a variety of shapes and sizes. They usually range about 10 to 20 micrometers in size. So they are about 10 times larger than our prokaryotic cells. Some of our eukaryotic cells can be quite large, and even some of the really big ones can be seen with the naked eye. Some of our amoebas, for example, are even a millimeter in size. Okay, so when we think about the evolution of life, we think about the origins of life, right? Life evolved on this planet billions of years ago, and it started out as a simple prokaryotic organism. And we call that simple prokaryotic organism the last common ancestor. All living things arose from this last common ancestor. Over time, different organisms evolved from that organism. So the first type of organism to arise was our bacteria, and you can see there on the left side of our screen here. They diverged first, and then we had the evolution of our archaea, and then finally our eukaryotes. When you look at this tree, you should see that your eukaryotes are more closely related to archaea than they are to bacteria. So the development of the modern eukaryotic cell occurred in a variety of steps. So we started out with this simple prokaryote, right? It did not have a nucleus. And the first step in the evolution of our eukaryotes was the development of that nucleus. So what we think happened is we had infoldings of the plasma membrane that eventually surrounded that DNA and became the nuclear envelope. Then we had the uptake of an aerobic bacterium into the cytoplasm of the cell. That aerobic bacterium lived symbiotically inside the cytoplasm of that cell and provided ATP. This process of an organism living within another organism is called endosymbiosis. So we had this aerobic bacterium that was living endosymbiotically, and eventually that gave rise to what we now know as the mitochondrion. Another endosymbiotic event that occurred is the uptake of a photosynthetic bacterium. And that photosynthetic bacterium became what is now the modern chloroplast. So we had two endosymbiotic events that gave rise to these two organelles, the mitochondria and the chloroplast. And now we have our modern eukaryotic cell. So that's what this looks like here. And it has a variety of internal structures to it. That is kind of the defining feature of our eukaryotes. So they have what we call organelles. Organelles are membrane-bound compartments within the cytoplasm of the cell. They have a very specific function. Each organelle does something a little bit differently within the cell. And we're going to talk about each one of them today. So here we have a list of all the different structures that we are going to discuss today. We will start with the structures that we find in our prokaryotic cells. So that includes the cytoplasm, the ribosomes, the plasma membrane, the cell wall, and the glycocalyx. And then we're going to talk about those structures that are unique to our eukaryotes. So that includes everything from the nucleus on down this list. So like I said, we're going to start with our common structures. So both prokaryotes and eukaryotes have cytoplasm, ribosomes, plasma membranes, and a glycocalyx, and then some of our eukaryotes have a cell wall. Now we're going to talk a little bit about the differences in these structures in our eukaryotes from that which we see in our prokaryotes. So first up is our cytoplasm. Just like in our prokaryotic cells, it is a dense fluid matrix that exists inside the cell membrane or the plasma membrane. It is the site of most of our biochemical reactions that occur within the cell. So most of those different chemical reactions that fuel living things occur right here in the cytoplasm. One big difference is that here in our eukaryotic cells, the cytoplasm is where we find our organelles. They're embedded in this matrix. Next up are the ribosomes. Ribosomes, remember, are responsible for protein synthesis. And just like in our prokaryotes, the ribosomes have a large subunit and a small subunit, except in eukaryotes, the entire ribosome put together is considered the 80S ribosome. Remember, in our prokaryotes, it is a 70S ribosome. Remember, S stands for Svedberg unit, and that reflects the size and the shape of the molecule. So eukaryotes have a slightly different set of ribosomes. Now, there are ribosomes that are found floating freely in the cytoplasm, and then you have some that are considered bound ribosomes that we see on the surface of organelles, specifically the endoplasmic reticulum. They're all kind of the same pool of ribosomes, though. So one place where we do see some similarities between prokaryotic and eukaryotic ribosomes is in the mitochondria and the chloroplast. Both of these organelles, which arose through endosymbiosis, have their own ribosomes, and these ribosomes are 70S ribosomes, so they look more like our bacterial ribosomes. Okay, just like our prokaryotes, eukaryotes have a plasma membrane. Remember, the plasma membrane, its function is to separate the inside of the cell from the outside of the cell. And it is composed of phospholipids and proteins. One big difference is that in eukaryotic membranes, we have these molecules called sterols, like cholesterol. They help to stabilize the plasma membrane, especially in organisms that don't have a cell wall. Now you'll notice in the plasma membrane it has kind of an interior side and an exterior side. The exterior side you might find glycoproteins or glycolipids, where you have chains of polysaccharides attached to the exterior side of the plasma membrane. You won't have that on the interior side of the membrane. So we do have some specialized lipids that we see in our eukaryotic membranes as well that we don't see in our prokaryotic membranes, things like sphingolipids. They're unique to our eukaryotes. And finally, one big difference is that our eukaryotes can participate in what's called bulk transport. That's endocytosis and exocytosis. So endocytosis is the process of bringing materials into the cell, and this is a type of energy requiring transport, right? It's a form of active transport, but here the membrane itself is used as a transport mechanism. So the membrane pinches off and forms these little bubble-like structures. We call them vesicles that then come inside the cell. So there's three types of endocytosis. The first is phagocytosis, and we will actually talk a lot about phagocytosis this semester because that's one tool that our bodies use to eliminate things like bacteria and viruses, is through the phagocytic pathway. But that's a process of taking in large particles from the outside. Penocytosis is a process of what we call cell drinking. It is where the cell forms little invaginations in the plasma membrane, little cup-like structures, and we take up whatever solute is in the exterior environment, gets captured inside the vesicles. The last type of endocytosis is receptor-mediated endocytosis. This is where proteins on the surface of the cell capture specific molecules. Those proteins are called receptors, and they bind those specific molecules out in the exterior environment, and then that triggers the uptake into the cell. So all three of these mechanisms result in the formation of these little membrane enclosed compartments inside the cell. Each one's a little bit different in what it's taking up and how specific it is. So again, your three types of endocytosis are phagocytosis, penocytosis, and receptor-mediated endocytosis. The opposite of endocytosis is exocytosis. So exocytosis is the use of these membrane-bound compartments, these vesicles, to release material out of the cell. So in this case, these compartments, these vesicles, fuse with the plasma membrane and they release material into the extracellular environment. And a variety of different materials can be released in this way. One example is insulin that is released from our pancreatic cells. When we eat a sugary meal, those cells are triggered to release insulin into the bloodstream through exocytosis. So that's just one example of that. In addition to exocytosis and endocytosis, eukaryotic cells also depend on simple diffusion, facilitated diffusion, and active transport. So all of those mechanisms for getting materials in and out of the cell are also found in our eukaryotic cells. Remember, our prokaryotes have that one specialized type of transport called group translocation. That's one thing they have that eukaryotes do not. Beyond the plasma membrane, we have a structure we call the glycocalyx or the extracellular matrix. This is going to be the outermost surface of the cell, and it consists of polysaccharides and proteins. And it's going to differ from cell to cell. It provides protection to our cells and adherence. It allows cells to attach to one another. Remember, the glycocalyx in our prokaryotic cells was our capsule or our slime layer. Here it's a little bit more varied in our eukaryotic cells, and like I said, it's going to differ depending on what type of cell we're looking at. So one of the important things that occurs with the glycocalyx or the extracellular matrix is that it allows for cell signaling in our eukaryotic cells and recognition of our cells. So for example, our red blood cells have a polysaccharide on their surface that's part of that glycocalyx that is responsible for determining self versus non-self, right? Remember the A, B, and O blood types? That depends on an extracellular polysaccharide that we find in that extracellular matrix. Some cells have a cell wall that comes before the extracellular matrix. So in our eukaryotes, we have two different types of cell wall. We have our fungi that have cell walls, and those fungi have cell walls that are composed of a molecule called chitin. And then our plants, they have cell walls that are composed of a molecule called cellulose. All right, that's going to be different than what we see in our prokaryotes, right? Our bacteria, their cell walls are composed of peptidoglycan. The function is essentially the same, however. It's there to provide structure and support to the cell. Now we are going to move on to structures that we find in eukaryotes that we don't find in prokaryotes. And the most important of these, of course, is going to be the nucleus. So it is usually the largest organelle in a eukaryotic cell. It's usually the most obvious organelle in a eukaryotic cell. And it separates the DNA from the cytoplasm. It protects the DNA. Remember, we've got all those biochemical reactions that are occurring out in the cytoplasm. We don't want those reactions to accidentally damage our DNA, so having a nuclear envelope, having a separate membrane around the DNA, helps to protect it and keep it from being damaged. So that membrane is actually a double membrane, so you actually have two phospholipid bilayers that encase the DNA inside of a eukaryotic cell. Within the nucleus, we have a structure called the nucleolus, and the nucleolus is responsible for ribosomal synthesis. So the nucleolus is really just an area that appears darkened under the microscope or using electron microscopy. You'll see a darker area inside the nucleus, and that is the nucleolus region. That's where we see ribosomal RNA being produced and those ribosomal subunits beginning to be assembled. We don't have a nucleolus in our prokaryotes. Remember, in a prokaryote, they don't have a nucleus, but they have a nucleoid. So that gives us three terms that we have to remember that sound very similar to one another. You have nucleoid, that's strictly prokaryotes. Nucleus, that is the large structure that contains the DNA of the cell. And then your nucleolus is inside the nucleus, and that's where we have ribosomal assembly and ribosomal synthesis occurring. Within our eukaryotic nucleus, we have our chromosomes and our chromatin. Remember, a chromosome is just a single unit of DNA, and eukaryotes have multiple linear chromosomes. Now, when the cell is going about its daily business and is not in the process of cell division, those chromosomes are all kind of intermingled and overlapping one another. It kind of looks like a giant bowl of spaghetti. And what's happened in that case is the DNA is wound around proteins, but it's kind of loosely organized. That DNA organized around proteins is called chromatin. The entire segment of DNA is called a chromosome. Again, eukaryotes have multiple linear chromosomes, where prokaryotes have a single circular chromosome. Beyond the nucleus, we have a structure we're calling the endomembrane system. And this is a set of membranes that kind of continuously flow from one into the next into the next. And that it consists of the endoplasmic reticulum and the Golgi apparatus. And then these vesicles and lysosomes that bud off of these organelles. So the endoplasmic reticulum, the rough endoplasmic reticulum is the site of protein synthesis for proteins that are going to get secreted out of the cell. And the smooth ER is a place where we have a variety of different enzymes that serve functions like a drug detoxification and lipid synthesis. The Golgi is kind of like the mailroom of the cell. Proteins that are made in the ER are transported via vesicles from the ER to the Golgi apparatus, and then they're sorted and targeted for various destinations throughout the cell. Many of those proteins will then be secreted from the cell via exocytosis. One structure that I want to point out in this figure is the lysosome. Lysosomes are specialized vesicles that form from the Golgi that contain digestive enzymes within them. They are very important for breaking down materials inside the cell, but those digestive enzymes are separated from the cytoplasm so they can't damage the cell itself. Instead, they're just used to digest whatever's inside the lysosome. Again, we'll talk about lysosomes when we get to phagocytosis later this semester, because lysosomes play an important role in that process. The next organelle is your mitochondria. You probably remember the powerhouse of the cell, right? The mitochondria are the site of ATP synthesis in our eukaryotes. We talked about how they arose through endocytosis, through evolution, and they have their own DNA and their own ribosomes. And within our mitochondria, we have the reactions of the Krebs cycle and the electron transport chain that are responsible for making most of the ATP that your cell is going to need. So that's the main role of your mitochondria, is to help extract energy from the food that is taken in by the cell and convert that energy into ATP, the energy currency of the cell. It has a double membrane and it has its own DNA and its own ribosomes. Next up are the chloroplasts. Chloroplasts are the structures that are responsible for photosynthesis in plants and in some protists. They also have their own DNA and ribosomes. Again, they also arose through endosymbiosis. Peroxisomes are kind of a unique organelle. They got their name because they produce hydrogen peroxide, but they also play an important role in lipid biosynthesis. They contain an enzyme called catalase, which is responsible for detoxifying hydrogen peroxide, and they are involved in the breakdown of biological molecules. So they have a lot of different roles. The enzymes and the chemicals that are produced within peroxisomes are toxic to the cell. So again, you have that idea of containing those substances in a compartment that is separate from the cytoplasm, so you don't have those things damaging the rest of the cell. Next up is the cytoskeleton. The cytoskeleton is a complex network of protein fibers extending throughout the cytoplasm. These fibers provide structural support and allow for cellular movement. There are three types of cytoskeletal fibers: microtubules, microfilaments, and intermediate filaments. The thickest fibers are the microtubules. They are composed of a protein called tubulin. Tubulin proteins polymerize to form long, hollow, tube-like structures. These microtubules act like train tracks throughout the cytoplasm, allowing motor proteins to transport cargo, like vesicles, from one side of the cell to another. Microtubules also help separate the chromosomes during eukaryotic cell division, during mitosis and meiosis. The thinnest filaments are the microfilaments. These structures are composed of two actin filaments wound around each other. They are involved in the circulation of the cytoplasm, forming the cleavage furrow during cell division and in muscle contraction in animals. Both microtubules and microfilaments are dynamic structures that can be assembled and disassembled based on the needs of the cell. Intermediate filaments are a little bit different. These filaments are more permanent in the cell, and they are composed of a variety of different proteins. these proteins wrap around each other to form rope-like structures. They are found on the underside of the nuclear envelope, creating a structure called the nuclear lamina that supports the nucleus and helps protect the DNA. They create anchors between cells. These anchors are called desmosomes. They can also be found outside the cell in the extracellular matrix and are used to strengthen tissues and connections between cells. One important role of the cytoskeleton is to provide for cellular movement. Actin filaments allow for a type of locomotion called amoeboid movement. This is when the cells kind of ooze across a surface. The actin filaments grow on one side of a cell, pushing the cytoplasm forward, while they shrink on the opposite side, pulling the cytoplasm with them. Microtubules form the basis of cilia and flagella. These are hair-like structures that are surrounded by the plasma membrane. They extend outward from the cell surface. While their internal structures are the same, cilia are short, while flagella tend to be much longer. Usually a cell will be covered in cilia, but may only have a few flagella. Both microtubules and cilia have bundles of microtubules within them. These microtubules are arranged in a 9+2 pattern with a ring of 9 pairs of microtubules surrounding 2 central microtubules. Motor proteins crawl along these microtubules in this ring and cause the cilia and flagella to bend and flex. This results in a characteristic wave-like motion. Remember, prokaryotic flagella are made of a protein called flagellin and move more like a motorboat propeller. Here, your cilia and flagella move more like a whip-like structure or in a wave fashion. Okay, so let's take a quick moment to compare prokaryotes and eukaryotes. So first off, your size. Bacteria and archaea are much smaller than eukaryotic cells, about 10 times smaller. Bacteria and archaea range from about 0.5 to 1 to 2 microns in size, where eukaryotes are 5 to 20 microns in size, and some are even larger than that. Bacteria and archaea don't have a nucleus. Eukaryotes do. The DNA of our bacteria and our archaea is composed of one single circular chromosome in our prokaryotes, but in our eukaryotes is composed of multiple linear chromosomes. Bacteria and archaea, they divide through a process of binary fission, which means simply the cells copy their DNA and then they distribute that DNA to 2 daughter cells. Eukaryotes are different. Remember, eukaryotes have a nucleus, so there's more steps involved in cell division. That's where we end up with mitosis and meiosis. Our prokaryotes have cell walls. In bacteria, that cell wall is composed of peptidoglycan. In archaea, there's lots of different chemicals that make up our cell wall. Sometimes it's called pseudomurine, but there's lots of other molecules that can be used. In eukaryotes, not all eukaryotes have a cell wall. Those that do, the cell wall is either composed of cellulose if we're a plant or chitin if it's a fungus. Now for motility, our prokaryotes can use flagella, and that flagella is composed of flagellin and has that propeller-like motion, where our eukaryotes, they have flagella or cilia, which are shorter, and they contain microtubules, so they're constructed completely different from what we see in our prokaryotes, and they move in this wave-like motion. Eukaryotes can also move in that oozing-like motion called amoeboid movement. Bacteria and archaea do not have organelles, membrane-bound structures within the cytoplasm. Eukaryotes do. There's no endomembrane system in our bacteria and our archaea, but there is an endomembrane system in our eukaryotes. And then finally, The ribosomes of our prokaryotes are 70S ribosomes, and our eukaryotes have 80S ribosomes. So they have a different size and shape. Of course, our eukaryotes also have 70S ribosomes in their mitochondria and their chloroplasts, but remember, those have their origins in endosymbiosis. Okay, so that's a good summary of the differences between our prokaryotes and our eukaryotes. Now that we've talked about the different features of our eukaryotic cells and how eukaryotic cells compare to prokaryotic cells, I want to talk about the different types of eukaryotes. So there are four main groups of eukaryotes. We have our protists, our plants, fungi, and animals. We are not going to talk much about plants. Again, they are not generally pathogens, so we don't focus on those. But we will discuss protists, fungi, and animals. So protists, they are single-celled eukaryotes, all right? And it's a very diverse group. So we used to call it the kingdom Protista, but it turns out that these organisms really aren't evolutionarily related to one another. The one thing they have in common is that they are single-celled. We can divide them into two kind of broad groups. One group is the algae. There are photosynthetic protists. And then the other group is our protozoa, or our animal-like protists. Protozoa means first animals. That's what that name comes from. So briefly, algae, they are phototrophs. That means they make their own food through photosynthesis. They are responsible for producing most of the world's oxygen through photosynthesis. So algae are very important. But they're not usually pathogens. There are a few examples of algae affecting human health. For example, they may cause food poisoning. There's a type of food poisoning called paralytic shellfish poisoning, where toxins that are produced by algae are accumulated in the shellfish that eat those algae. And then when we eat those shellfish, we can get sick. So that's one example where algae can impact human health. Another way that algae can affect human health is through toxic algal blooms called red tide. So red tide occurs when pollution creates an environment in rivers and oceans, or even ponds and lakes, where there's high amounts of phosphorus and nitrogen that support the growth of algae. Those algae then in turn can create a toxic environment for the organisms that live in that water. The algae can create an environment that can harm the fish and other wildlife that live in those environments, and the algae themselves and the byproducts of their life can actually create an allergic response in humans. So you've probably heard about that here in Florida because we've had kind of recurrent red tide over the last several years. Okay, so I really want to focus more on protozoa. Protozoa are single-celled. They have all the major organelles except for chloroplasts. They have cytoplasm that is divided into two parts. You have the ectoplasm and the endoplasm. The ectoplasm is usually clear, and it's usually the area of the cytoplasm that's responsible for locomotion, for feeding, and protection of the cell. And then the endoplasm is where you will find the nucleus and the mitochondria, and it's kind of granular in appearance. Protozoans do not have a cell wall, but some may have what's called a pellicle. A pellicle is a protein coating on the outside of the cell that can serve as a type of protection for the cell. These organisms are heterotrophs. Heterotrophs mean that they eat a variety of different things to provide them with energy and nutrients. Protozoans can form what's called a cyst. A cyst is a dormant stage, very similar to what we see with our bacterial endospores. They're highly resistant to chemicals and radiation and heating, and they're metabolically inactive. However, they can be activated into the feeding stage, and that feeding stage is called a trophozoite. Protozoans can get around in a variety of different ways. Some protozoans are motile, some are not. Those that are motile, that can move, can use cilia, like we see in the Ballontidium coli trophozoite in the first picture here. Some may use flagella. We have Trichomonas vaginalis here in the middle that has several flagella on one end there. And then some can use pseudopodia, right? That oozing-like motion. This is an amoeba that you see down here on the end, Entamoeba histolytica. Protozoan life cycles can be pretty simple. So you may have a protozoan like we see here. This is an amoeba that it has a trophozoite form, the feeding form, and then it has the cyst form, and it just cycles between the two stages. That's a pretty simple protozoan life cycle. Protozoans may reproduce asexually like we see here, or they can reproduce sexually. So this next life cycle I'm showing you, this is also a protozoan life cycle, but this is the protozoan life cycle for malaria, and you can see there are many different stages. And in this particular life cycle, you have both sexual reproduction and asexual reproduction. So it's a very complicated life cycle. So protozoans can go either way, it just depends on the organism. These are some examples of protozoan pathogens. Ballontidium coli, that ciliated parasite that I showed you, that is a organism that causes some gastrointestinal disease. You have your flagellates, so Giardia lamblia, which I know we talked about in our first lecture. That is a flagellated protozoan. Trichomonas vaginalis, which you saw the picture of just a few moments ago. That is the only STD, or sexually transmitted disease, that is caused by a eukaryotic pathogen. We have a couple of diseases down here, leishmaniasis and trypanosomiasis. Those are tropical infectious diseases that are transmitted by bugs. Again, these are flagellated protozoans. Up at the top here on the right-hand side, we have amoebas. So Entamoeba histolytica is an intestinal amoeba. Naegleria fowleri is one that you've probably heard of living here in Florida. That one is the amoeba that can get up into the nasal passages from freshwater environments and can grow in the brain and cause pretty serious disease and usually death once it gets there. And then we have a group of protozoan pathogens that are non-motile, that don't move, that we call apicomplexans. So we have Cryptosporidium. That's a very common parasite that causes diarrheal illness. Toxoplasma gondii is associated with cats, and most of us are infected with that one without even knowing it. And then finally, your Plasmodium species, which are your malaria parasites. So there's lots and lots of different protozoan pathogens. These are just a few examples of those. Fungi are the next group of eukaryotic organisms, and they are divided into two groups. The macroscopic fungi, that includes mushrooms, puffballs, etc. We don't talk about those in this class. And then we have our microscopic fungi. That includes molds, which are multicellular, and yeasts, which are unicellular. All of our fungi are heterotrophs, and some are parasites, where others are saprobes. So a saprobe is an organism that lives off of dead or decaying material, and a parasite is an organism that lives off of another organism while it's still alive. So fungi can live either way. They can be a saprobe or they can be a parasite. So for our multicellular fungi, they have structures called hyphae. Hyphae are these long filamentous structures that consist of multiple cells, and they have a cell wall that is composed of chitin. So here on the left of your screen, you can see your hyphae. They make up the body of your mold, and that body is called a mycelium. It's that fuzz that you see growing on your moldy bread, for example. Yeast are unicellular fungi, but they can form chains of yeast, and those chains are called pseudohyphae. Our fungi, when they can reproduce sexually or asexually. Our yeast, they primarily reproduce asexually by budding. A cell will bud off of another cell through mitosis. Or you may have with your molds the hyphae breaking off or fragmenting, and then they move to another location or carried to another location and will continue growing there. Or you may have spore production. So what you see in the picture below is some fungal spores that are produced by a mold. These are very different than our endospores in our bacteria. Fungal spores are lightweight, they can travel long distances, they can be found in the air, but they're not as hardy as your endospores. So fungi with regard to human health, most fungi are free living. Some are pathogenic. Those that are pathogenic, they are pathogens either as what we call true pathogens. We encounter them in the environment and they can cause disease in humans. We call those environmental pathogens. We have those that are hospital-acquired pathogens. So there are some fungi that are unique to the hospital environment, particularly in patients that are on respirators or ventilators. And then we have what are known as opportunistic pathogens. Those are fungi that cause disease only under the right circumstances. Usually when a patient is immunocompromised. That might be because they have HIV, or they're undergoing chemotherapy for cancer, or they're a transplant patient. All of those situations result in immune systems that are suppressed, and that can allow for some organisms to grow that wouldn't normally grow in the body. So this is just some examples of fungal diseases. A very common fungal disease is ringworm. It is also called tinea, athlete's foot, or jock itch. And you can see it's a skin infection. It can cause discoloration and redness of the skin. It often forms that kind of ring-like shape, which is where it got the name ringworm. It doesn't involve a worm at all. It's a fungal infection. Here's another fungal disease. This is candidiasis. Candidiasis is thrush or can be a yeast infection. So what you're seeing here is an individual who has an overgrowth of yeast in their mouth. It looks like maybe it's a baby. And you can see that those white patches on the inside of the mouth. A yeast infection can be a infection of the vaginal mucosa that can occur when the natural microbiota that are present in the vagina are disrupted. So oftentimes women who are on antibiotics, for example, can acquire a yeast infection. Candida is an organism that naturally occurs in our bodies, but when it overgrows, it can cause disease. And then another fungal disease is coccidiomycosis. This is an environmentally acquired pathogen. This is a pathogen that is found in the soil throughout the southwestern United States. So you can see in this map, you can see the areas in orange. Those are all places where the organism has been isolated from the soil. If individuals breathe this organism in, it can cause a respiratory disease called valley fever. It's not common here in the southeastern United States. Okay, our last group of organisms to discuss are my favorites. These are the animal parasites, or animals that cause human disease. So this category is also called our helminths. These are our worms. You have your cestodes, which are tapeworms, your trematodes that are flukes, and then these two types of worms are flatworms. And then finally we have our nematodes, which are roundworms. So all of these are multicellular organisms that can cause disease in humans. And they have various organs within them and organ systems, digestive tracts, reproductive systems, nervous systems. It depends on the type of worm that we're looking at. So they can reproduce sexually or asexually. The cestodes are tapeworms. This is a great example of a tapeworm. I think this one in the picture is about 8 feet long. So tapeworms are these flat tape-like organisms. That's where they get their name from. And they are segmented worms. So the segments, and there's a picture of one off here to the right, they're called proglottids. You don't have to memorize that. But the proglottids are individual segments of the worms, and each segment contains reproductive organs. So that structure that is colored in dark in that image is actually the ovaries of this particular worm. These are egg-making machines. So one thing that's unique about our tapeworms is that they do not have a digestive tract. They absorb nutrients from their host, from the organism that they're living in. This particular tapeworm lives in the large intestine, and it absorbs whatever you're eating. It is hermaphroditic, meaning that it has both male and female components, and it reproduces sexually. It also can reproduce asexually. The next group is our trematodes. These are our flukes. So these are also considered flatworms, although you can see in this picture, this one's got kind of a curved appearance to it. That is because that is two worms in that picture. That is a male and a female worm that are kind of nested one inside the other. The female is actually the larger of the two. That particular trematode is called schistosoma. And these ones mate for life. So that little male worm sits inside the groove of that female worm for the rest of its life, and they are constantly mating. So these worms are non-segmented, so you don't see those proglottids like we saw in our cestodes. They do have a rudimentary digestive tract. And although this one in the picture is dioecious, meaning it has male and female worms, most of your trematodes are going to be hermaphrodites. They have both male and female parts. They have very complex life cycles. And they reproduce both sexually and asexually. They have many different hosts. So what that means is there's different organisms that they live within. So many of our flukes will live within humans, and then their eggs will be released into a freshwater environment through sewage, and the eggs will hatch in a freshwater environment, and then will eventually find a mollusk or a snail to live in, and then complete part of its life cycle there before it then continues on with its life. So that's multiple life stages. Now, the host that is responsible for the sexual reproduction of the parasite is called the definitive host. So this is the life cycle of that schistosoma parasite that I was showing you. It's really complicated. These worms actually live in the veins surrounding either the large intestine or the bladder. So they live kind of in the mesenteric veins. The adult worms live there. They mate. They produce eggs. Those eggs actually are very sharp and pointy. You can see some of them down here on the bottom with these pointy little, they're called spicules on them. And they actually will be embedded in the tissues around those blood vessels. And eventually they penetrate either the large intestine or the bladder, where then they're released out into the environment in human waste. Those eggs will hatch, then the first larval stage will emerge, and then eventually find a snail to penetrate. It'll develop further in the snail. You might have some asexual reproduction within the snail, and then you'll have another life stage that emerges until it enters into the human. So it's a very complicated life cycle, multiple hosts. The next group of helminths is our nematodes. So we've talked about our cestodes, which are tapeworms, our trematodes, which are flukes, and then nematodes are roundworms. And they are also non-segmented. So the only one that has segments is going to be those cestodes, those tapeworms. They have a durable cuticle on the outside of them. That's a thick layer of protein that helps to protect the worm. And they have a complete digestive system with a mouth and an anus. So they have a complete digestive system. They have a well-developed nervous system and a musculature system. And they can be male or female. So they can be what's called dioecious. This particular worm in this picture is a worm called guinea worm. It has a really fascinating life cycle. I'm going to show that to you here in just a second. But what you're seeing in this picture is that the female worm, when it's ready to release her larvae, she actually forms a blister on the host, and that blister is usually on the foot or the leg, sometimes the hand or the arm, so it's on the extremities. And then when that blister enters a freshwater environment, it bursts open and it releases the larvae out into the environment. So this is what that life cycle looks like. Again, it's a little bit more complicated than what we see with, your protozoans, maybe that have that simple cyst and trophozoite form. Here we have the release of the first stage larvae from a blister on a foot. So these are people who are walking into what's called a step well to get their drinking water. They inadvertently release the larvae into that water. The larvae actually will then enter into copepods and will exist within copepods. And it's when we drink the water containing the copepods that are infected that we get sick ourselves. And then the parasite will actually migrate throughout the body, throughout the connective tissue, until ultimately it finds that location, usually in the lower extremities. So the only way to remove this worm, I'm going to go back to this picture for just a second, is to carefully pull on it and wind it around a stick a little bit each day. You can't pull this worm all out at once. If it breaks, that can cause a lot of problems. So you have to be very careful about the way that it's extracted from the body. This one's really an interesting one to talk about because it is almost completely eradicated from the human population. Because it's found in freshwater environments and drinking water, and it is in copepods, which are easily removed through filtration, we are able to eliminate this organism from the human population by simply providing access to rudimentary water filters, right, that can then remove the parasite. So that one's a good success story in terms of public health. So that is our eukaryotic parasites. I hope you found this last little bit interesting as we talked about some of the eukaryotic parasites that cause human disease.